Hello, my name is Nur Fatima binti Bahrudin with number matrix BS24110016. I want to present about practical test that is demonstrate dissection to obtain NNV infected organ and preserve the sample for histology. This is my introduction. Viral nervous necrosis (VNN), also known as viral encephalopathy and retinopathy, which is VER, is a serious viral disease affecting marine and freshwater fish. Is it caused by the nervous necrosis virus (NNV), which mainly attack the brain, spinal cord, and retina. Infected fish often show abnormal swimming behavior, loss of appetite, and dark body color, and high mortality, especially in larvae and juvenile fish. Histological examination is a standard method for diagnosis VNN. Therefore, proper tissue collection and fixation using 10% formalin are important to preserve tissue structure for microscopic analysis. So our objective is to learn the correct procedure for surgically harvesting NNV infected organ and preserving tissue sample for histological analysis. For material requirement, we use fish suspected of NNV infection, surgical scissor, forceps, dissection pins, 10% natural buffered formalin, which is NBF, sample bottle, and disease board. For step 1, which is the preparation stage, all dissection instruments were first prepared and sterilized. Before handling the fish, I spray 17% ethanol on my hands, the fish surface, and the dissection tray to reduce contamination and maintain aseptic condition. This step is important to ensure that the tissue sample collected are clean and sustainable for histological examination. After sterilized, the instruments were arranged properly and ready for the dissection process. For step 2, an internal examination was carried out to observe the physical condition of the fish. The fish was carefully inspected for any visible clinical signs associated with VNN infection. During the observation, dark body coloration and eye abnormalities was recorded. These signs are commonly associated with viral nervous necrosis and provide important preliminary information before further tissue examination and histological analysis. Next is the collection of target organ, but before we collecting the target organ, the dissection instrument will sterilize again to prevent contamination. The forceps and all equipment that we use were immersed in 17 ethanol and the frame sterilized using a burner to ensure they were free from microorganisms. After framing, the forceps were allowed to cool before being used. Once the instruments were ready, the brain and eye tissue were carefully collected to histological analysis. After collection, the tissue was trimmed into small pieces of approximately 0.5 to 1 cm in thickness. This step is important because smaller tissue samples allow the physics-active solution to penetrate more effectively and evenly throughout the tissue. Proper fixation helps preserve cellular structure, prevent tissue decomposition, and ensure that any lesion or pathological change caused by VNN can be clearly observed under a microscope during histological analysis. For this step, the target organ, which were the brain and eye, were carefully collected from the fish. This organ was specifically selected because VNN primarily infect the central nervous system and retina, where the virus concentration is usually the highest. As a result, the brain and eyes often show the most obvious pathological change and are considered the most important tissue for diagnosing VNN through histological examination. For step 4, the brain and eye tissue sample were immediately immersed in 10% natural buffer formalin to preserve the tissue structure and prevent decomposition. The tissue were placed in a 1% tissue to fixative ratio and completely submerged to ensure proper fixation. The sample were then left at room temperature for 24 until 48 hours to allow the fixative to penetrate the tissue before histological analysis. Finally, the sample via were tightly kept sent to the histology laboratory for histological examination to detect tissue change caused by VNN infection. That's all from me. Thank you.